Hello friends, in this lecture we will be discussing the components of a scientific paper, and of course these are the components that are supposed to be there even in a thesis. So whenever you are writing a thesis or writing a research paper, especially if that is of original research, then what should be the components? This is what we will be discussing in this lecture. So a typical scientific paper, especially the original research paper, consists of certain components. So first one will be the title, next is authors and their institutional affiliation, third one is abstract, then comes keywords, then introduction, material and methods, results, of course, which include tables and figures, discussion, acknowledgement, and lastly the references. So these are the essential components of a typical original research paper. Now title. How should one write the title of a paper? So title should be complete, specific, and should cover the central idea. The central idea of the research should be summarized in the title. Should include the key methods such as survey or double-blind randomized placebo-controlled trial or observational study, etc. These are the terms that must be there in the title. Independent and dependent variables must also be included in the title. For instance, the effect of maybe Amavatari Rasa in Amavata. So Amavata and Amavatari Rasa. These are the two variables that must be there. Name of the species, if not human. The species name also should be included. Do not use abbreviations, jargon, chemical formulae and series identifiers. Don't use anything that one is not able to understand. So the title should convey the central idea of the entire research work. While identifying the central topic, you can be creative, of course. For instance, the immunomodulatory, neuroprotective and longevity enhancing effect of 𝐴𝐵𝐿𝐼 officinalis in mice is always better than evaluating the Rasayana effect of 𝐴𝐵𝐿𝐼. Because when you use the terms Rasayana and 𝐴𝐵𝐿𝐼, a reader who may not be conversant with the Sanskrit language and with Ayurveda may not be able to understand what your study is all about. Neither it reveals whether the study is a clinical study or an experimental study carried out in animal model and so on. So this is incomplete title, and also it doesn't give a clear idea about the study. Suppose if you are writing a review paper, then you can use your creativity. For example, how did Sushruta treat typhoid is better than the concept of 𝑗vara in Sushruta Samhita. So the same title, same topic, but if it is converted into attractive and interesting title, it is always better. So title for clinical research articles must include the setting, the location, the patients, what was studied, intervention, the treatment, what was given, comparator, what was the control group, whether it was positive control or placebo control or no intervention, and so on. Endpoint, what was the outcome of the interest and what study design was employed. All these details should go into the title. So that is how a good title is written. While writing the title, please avoid institution's name or maybe number of cases in the title. For example, outcomes of Panchakarma, the Mumbai Ayurveda hospital experience, or outcomes of 𝑘𝐻𝑎𝑟 trial in Amavata, a review of 275 cases with 5-year follow-up. This is not how a good title should be written. Similarly, interrogative titles, the titles that end with a question mark, should usually be avoided Measurement of colonic polyps by radiologists and endoscopists. Who is most accurate? This is not the ideal title. Usually such interrogative titles are reserved for editorials or discussion articles. Also avoid declarative titles. For example, the grapeseed extract prevents skeletal muscle wasting in interleukin-10 knockout mice. Here what is being written is the results instead of writing what was studied. So don't write the results instead of writing what was studied. Then comes running title. So running title is a short title of the article which appears on the top of the other side of the journal pages, and usually it is short and up to 50 to 75 characters, and use independent and dependent variables. You can omit species and you can be creative and adhere to the journal guidelines while writing the running title. After you have written the title, the next is the abstract. Of course, title and abstract are usually written at the end of the paper, not at the beginning, but I am following the sequence as it appears in the published paper. So title is the most frequently read part of any published paper, and abstract is the second most frequently read part of any published article. So reader decides to go through the paper only if he or she finds the abstract interesting. Hence, the abstract should be brief. It should state the problem and hypothesis. It should state the methods, maybe random control trial or observation study or whatever. Should summarize the results. Should include the inferences of the study, and you should avoid known or generally accepted facts in the abstract. So these are the points that one has to keep in mind while writing the abstract. It has been suggested that about 10% of the abstract should be dedicated to introduction or background, where you will state what was the purpose of the study. Then about 30 to 35% of the abstract should be dedicated to material and methods. What was the study design? What were the techniques employed? And what statistical methods were used? Then around 35 to 45% of the abstract should go for results. What are the most important findings? This is what you should describe in the results section of the abstract. Then conclusions, usually 20 to 25% of the abstract. Why are these results important? This is what you should state. So the goal of an abstract is maximum information in minimum space. This is the goal of a good abstract. And there are two types of abstracts. One is structured and another one is non-structured. In structured abstract, usually the headings are given to identify the content, and it follows IMRAD format, that is introduction, methods, results and discussion format, typical in North American journals, and usually the word limit is around 250 words. In unstructured format abstract, the entire abstract is arranged in one full paragraph. Of course, this too follows IMRD format, but no separate headings are given. This is the only difference. This is typical for European journals, and usually it allows for around 150 words or so. Of course, this word limit is not universal. It may differ from journal to journal. What makes an abstract good? When can you say that an abstract is good? If it follows the guidelines of the journal, it should be a piece of information which should be understandable in isolation, that is stands alone, and it is an independent unit of information, and it should be accurate, check for inconsistencies and omissions. It should be readable and coherent, and it should include the specific data, which is very important. So if your abstract includes all this, then it should be a good abstract. So these are the 6 steps which are generally advised while writing the abstract. So identify the guidelines of the journal, highlight the key features, insert the sentences into abstract format, write, revise and condense, edit the sentences and words, and check the final. This is a very commonly written kind of abstract. We will see how this abstract can be rewritten, or whether it is good or can be kept as such. So let me read it. Ayurveda is one of the most ancient healthcare systems in the world. The term Ayurveda consists of two words, Ayu and Veda. Ayu means life and Veda means knowledge. Therefore, Ayurveda means science of life. In Ayurvedic literature, immunity has been described as ojas. This is of two kinds, para and apara. In the recent years, immune system related disorders are being increasingly recognized, and the role of immunomodulatory drugs is being highlighted. In Ayurveda, Rasayana drugs are described to have immunomodulatory effect. The present study aims at evaluating the role of Amalaki as an immunomodulatory agent in an animal model. Details have been explained in the paper. So this is a typical abstract that we often receive as journal editors, and especially this is a very commonly written style in which the abstracts are submitted for conferences. But actually this is not the right way of writing a good abstract. In fact, this is the only sentence that should be retained, and all the other sentences are to be removed from this abstract to make it a good abstract. The present study aims at evaluating the role of Amalaki as an immunomodulatory agent in an animal model. This should be the first sentence of the abstract, and the details of the study should be there. Next is keywords. So ideally, keywords should not be from the title and abstract, because the title and abstract, they are already indexed with most of the indexing databases. So all the words that appear in title and abstract are already included. So you give additional words which a reader might use while searching for your paper, and they should not be the same that are already there in the abstract and title. So databases use keywords for indexing the literature, and users search a database by keying in the keywords. So more diversity in the keyword selection means more visibility. This is the key point. And whenever possible, use mesh terminology or mesh thesaurus, that is medical subject heading, if not sure of keyword usage. For instance, autistic disorder is a mesh term, whereas autism is not a mesh term. So instead of autism, you can go for autistic disorder. So these are a few examples where you can use alternative terminology. For instance, if you have used the word Ayurveda in the title and abstract, then go for traditional Indian system of medicine as a keyword. Keyword need not be a single word, it may be a phrase as well. Lung function tests. Instead of lung function test, you can use respiratory parameters. Or Prakriti, you can replace it by Ayurveda constitution. Winter blues, you can use it instead of seasonal affective disorder. Insomnia, you can use sleep disorder. So this is how you can use multiple different words to mean the same thing, and thus increase the diversity and increase the visibility of your article. So medical subject headings have been developed, and this is called controlled vocabulary for biomedicine, and the purpose of mesh terms is to provide uniformity and consistency to the indexing of the biomedical literature. If you go for PubMed and if you search for different thesaurus, you will get this mesh thesaurus or mesh option where you can search for the mesh terminology. Now, IMRD, that is Introduction, Methods, Results and Discussion. This is also called IMRAD structure. A stands for AND. So IMRAD structure was originally developed by Pasteur and established by American Standards Institute in 1972, and it serves as an organizational format. And as it is said, IMRAD is a format, and of course each paper would differ. So any original paper will be organized and should be organized in IMRaD format. So what does it mean? What should introduction be about? Whenever you are doing some research, you ask some question, right? And that is what should be written in introduction. What question was asked? Why was it asked? Material methods should include attempts to answer the question. How did you try to get the answer to the question that you asked? Results should contain the data that you have obtained. So obtain and compile the data. This should be the content that should go in results. And then discussion, answer the question. So what is the answer that you got out of your research? So this is basic meaning of IMRD. So introduction, what should be there in the introduction? Based on what was known and unknown, why did you do this study? This is how you should usually begin your introduction. State what is known, then state what is not known, and then also tell the reader what your study will answer, right? So what was the research question? What was the tested hypothesis? And what was the purpose of this research? So all these questions should be answered in the introduction section. So clearly state the problem or objective, ensure that the problem is specific, important and relevant, state the previous work related to the problem, state the lacunae in the current understanding of the problem, and explain clearly how is your work different from the one that already exists. Then second one is statement of hypothesis. So first one was statement of the problem. The next one is statement of the hypothesis. So state the hypothesis clearly. Ensure that the existing literature supports your hypothesis. State briefly the methodology followed in the study. So these are the steps to follow while writing the introduction. Begin with what is known. State what is unknown. State what did you hypothesize, and end with what your study will answer. So this is how you can arrange a good introduction of a paper. So these are the common problems that we encounter as editors. Research question is often not specific, or sometimes multiple unrelated questions are addressed simultaneously. Key arguments are not backed up by appropriate references. The stated aim of the paper is vague or biased. For example, establishing the effect of 𝐴𝐵 on immunological parameters. This is a biased statement because you already want to establish something, right? Rather, the term that is ideally suited for such study is evaluating the effect, right? So the introduction is too long and unnecessarily reviews the entire literature is another common problem that we encounter. Too many objectives and inflated statements are very commonly observed in introduction. So this is what you should avoid. Next section is methods. When, where and how did you do this study? You should describe it in sufficient details. What materials were used? What instrument did you use? What protocol was used? Right? So what were the standards that were followed? Right? Who was included in the study? Study groups, maybe control group, patients or animals, etc. What statistical methods were used? So all this should go in methods, and methods should be sufficiently detailed. And usually methods section is the easiest one to write because this is how you have done the study. So whatever you have done, you should write. And please keep in mind that methods are to be explained with sufficient details, because if another researcher wants to repeat the study, he should be able to do the study without having to refer to some other material. So reproducibility is one important aspect of a good research, and if a research is not reproducible, possibly it is not a good research. And reproducibility can be ensured only when sufficient details of methods are provided in detail. Also state clearly the study settings, population, sampling strategy, instruments, data collection methods and analytical strategies. Provide justification for the appropriateness of the methods employed if unconventional. If you have used some unconventional method, please tell the reader why did you use those unconventional methods and what justification that you can give. Provide the readers with sufficient details so as to study may be reproduced. Ensure that the technical terms really match with the actual methods followed. For example, randomly selected means it should be randomly selected, right? Randomization is a technique in statistics and it has got a specific meaning. For example, in a method section, you should write the methods adopted for the study like population, specific criteria considered for inclusion, sample frame, sample, preparation of the preliminary questionnaire if it was survey, validation of the questionnaire for its reliability and consistency, collection of the data, response rate, and so on. Now these could be the different kinds of studies in observational category: case control study, cross-sectional study, longitudinal study, cohort study, panel study, and so on. Whatever kind of study that you might have carried out, those should be clearly stated and appropriate study designs should be followed and completely described. Experimental and clinical trials. In case of experimental and clinical trials, whether the clinical trial was blinded, was it double blinded, single blinded, and so on, was it open label trial, was it adaptive clinical trial or non-randomized clinical trial, and so on. So all these details should be there in the methods. So these are the recommended readings: consort statement, maybe reporting randomized control trials of herbal interventions, an elaborated consort statement. The consort extensions are there. Standards of reporting Ayurvedic clinical trial. Is there a need? This is an article published. You can refer to this. Efficacy and safety of Ayurvedic medicines, recommending equivalence trial design and proposing safety index. Drug and Cosmetic Act 1940, Drug and Cosmetic Rules, 4th Amendment, WHO General Guidelines for Methodologies on Research and Evaluation of Traditional Medicine. These are certain guidelines that one needs to refer. A few crucial details should be included in the method section. For example, sample size calculation. How did you calculate the sample size? This should be there. Method of random sequence generation. Details of blinding procedure. Preparation of the compound drug. Right? Simply writing that Kukkutaputta was given or Bhavana was given is not sufficient. Right? How the Kukkutaputta was given, right? What was the setting? All this should be there. Identification method of raw herbs. Voucher specimen retained or not. Rationale for placebo controlled trial. Clinical trial registration number. Ethical clearance. Informed consent. All this should be there in the methods section. So these are some important guidelines one needs to go through. ICMR ethical guidelines, international guidelines for care of animals, international guidelines for research involving humans, medical ethics manual, WMA declaration of Helsinki, ethical principles for medical research involving human subjects. Then there are some important guidelines, maybe START statement, consort extensions, strobe statement, prisma statement. For different kinds of trial designs or study designs, you have got different statements. All these different statements or guidelines can be obtained by logging into this Equator network. Equator network is one place where you get all kinds of different guidelines which are applicable for different study designs. Methods could be innovative. This is one example to say that you can use innovative methods. Double-blind randomized controlled pilot study comprising classical Ayurvedic medicine, methotrexate and their combination in rheumatoid arthritis. One needs to go through this article and where they have retained the individualization aspect of Ayurveda, but still they have been able to successfully conduct a double-blind randomized controlled study. So common problems that are encountered when it comes to methods is like methods, interventions and instruments are not described in sufficient detail. so that the study can be reproduced. So this is very important. Like 𝘕𝑦𝘗𝑗 was performed once daily. If you write this, it is often not sufficient. What time of the day, for what duration, what was the clinical setup, all this needs to be described. Coconut oil was used as placebo in control group, patients with atopic dermatitis. So in such cases you need to justify, why did you choose coconut oil? Has coconut oil been proven to have no healing effect or no aggravating effect in this condition? Then only you can go for coconut oil as a placebo. Otherwise, you cannot possibly. Placebo was given in the form of a capsule. What did that capsule actually contain? So inadequate designing or description of the study protocol is often a problem. What was the qualification of the physician who prescribed the control drug, phenytoin sodium, in apasmar maybe? Were the antibiotics given along with the trial drug, some ointment in the case of infective ulcers? Often, no control group in clinical trials involving very common conditions such as common cold. So in such cases, at least one has to have control group. What concentration of the solvent was used for extraction? What method of extraction was employed? All these details are often missing. How was the questionnaire to assess Prakriti constructed? How was it validated? And so on. All these details are required. Vaguely defined identification procedure of the herb. This is again a very common problem. Mr. XYZ, a botanist of the institute, identified the herb is usually not sufficient. Rather, you should write whether the morphology or HPTLC, chemoprofiling or DNA fingerprinting or whatever other methods that were employed to identify the drug. Those methods should be described. Simply mentioning a botanist's name is often not sufficient. No mentioning of the source of procurement. No mentioning of whether the voucher specimens have been preserved or not. What is the reference number of the sample, etc. All these are often missing. Control group is not comparable with the experimental group. This is again another common problem with the methods section. When you have a control group, it should be comparable to the experimental group. For example, details of the procedure involved in Varahaputta, the temperature range, duration, instrument used, etc. are often not given. A photograph of finished product is desirable along with the photograph of method of preparation. Where was it prepared? Where the good manufacturing practices followed. For instance, if you are preparing some bhasma, then all these things or all these aspects should be taken care of. Then statistics. Often many people think that statistics is jargon or redundant, but statistics is very essential part of any research article, and whenever writing the significance, you should take into consideration the difference between statistical significance and clinical significance. Though sometimes you may get some statistical significance as per some statistical tests, this may not translate always into clinical significance. So these terms such as significance should be used very carefully. Statistics is usually a very important component of any quantitative research. All inferences depend on proper interpretation of statistical results. If there is a real clinical effect, statistics will reflect it. And if the inferences drawn are not according to the statistical results, your paper is likely to be rejected. So use statistics appropriately and maybe take help from a statistician. Right? State what test was used to determine whether the difference between two or more groups was significant. Provide all details such as values of test statistics and confidence interval along with p-value. Ensure that the degrees of freedom are consistent with the sample sizes stated or the particular analysis carried out. So all these should be taken care of. Clearly state whether the number given after mean is standard deviation or standard error. Ensure that the numbers add up correctly. The sum of all the subgroups should equal the size of the full group. Define the technical terms that are specific to the methodology. if the methodology is unconventional. So what I would suggest is, please consult a good statistician at the stage of protocol designing, because statistician would be able to tell you how to collect the data, how to enter it on the computer, right? Consult a good statistician during data entry. Consult a good statistician while analyzing the results also, because statistician would tell you which kind of a test to be applied in which instance. Consult a good statistician while writing the paper. Again, during writing the paper also, better you consult a statistician, and it is always better to have a statistician as a co-author, so that any reviewer comments can be addressed. Next section is results. So in the results section, what you are supposed to write is, what answers did you get, and was the tested hypothesis true? So the answers to these questions should be there in the results. Ensure that the results section is straightforward and factual. Ensure that all results related to the primary question are given in detail, including simple counts and percentages. Ensure that the details like time frame, response rate, sample characteristics, etc. are stated, and also clearly state the key findings and secondary outcomes, if any. So this is how you have to write the result section. While constructing tables, again you need to give some extra thought while writing the table. The table should be understandable even without the text, self-explanatory, and simplify and condense the tables wherever required. Merge 2 or more tables if necessary and if possible. Remove any table if the data is not related to the problem. And stick to the general rule, like one non-text item per 1000 words. So non-text item would be either a table or a figure. So do you find this table alright? See, fundamentally this table is not alright, because we don't know what is group A and group B, what is BT and what is AT, and there are some factual errors as well, like urine microscopy parameter, right? Albumin and sugar are not the urine microscopy parameters. So this is not a good table, right? So provide sharp figures with lettering proportionate to the size of the figure. Use legends to explain the figures. Ensure that there is no manipulation in the photographs, intentional or unintentional. To reproduce a figure from any other source, you need license or permission from the original author or publisher, and the source should be cited. If your study is based on some visual parameter like histopathology, you must provide the photographs. Is this alright to use this photograph in your paper? Better to avoid it, because it reveals the identity of the patient. Maybe at the maximum this portion, this is the portion that you can use. Common problems. Results are reported selectively. Example, percentages without frequencies, p-values without measures of effect, and so on. Incomplete statistical analysis. For instance, p less than 0.001 derived after applying ANOVA, but no post hoc tests are applied. The repetition of results both in table and in text is unnecessary. Unrelated tables and figures are provided. Inadequate or inaccurate interpretation of statistical results. No test applied to compare the means, or no details about the mean readings that were compared. For example, while comparing the mean readings of blood pressure before treatment and after treatment in 2 or 3 groups, right? Which one is before treatment, which one is after treatment, right? All these details are often not provided. Redundant details about sample characteristics like religion, socio-economic status, education, marital status, etc. All this may not be required in a study that has got nothing to do with this, right? Conversion of actual numbers into groups and categories, and then allotting your own scores. This is very often committed mistake which should be avoided. Not following the standard criteria to assess improvement in clinical condition. Again, you cannot use your own criteria to assess the clinical improvement. Using wrong words to indicate change. You cannot use relief or improvement to just indicate the change, right? Actual values are not given on graphs. Again, the graphs should always contain the actual numbers. In this table, for instance, number of patients in different groups is given instead of mean and standard deviation of the observed parameter. So of course, whenever it is possible, you should go for mean and standard deviation instead of going for simple numbers. No actual values in the graphs. See BMI, weight, BMI and body fat percentage. No actual values are given in the graphs. Here, the authors have converted the continuous data into discrete data. Serum cholesterol more than 240 mg per dl, 200 to 239 mg per dl, less than 200 mg per dl, and then they have classified or allotted some scores, and this is totally done arbitrarily, and this should be totally avoided. It should always be avoided, because when you are having the actual serum cholesterol concentration with you for every patient, it is always advisable to go for mean and standard deviation and not for converting them into discrete data. Not following a standard criterion, right? So complete remission, marked improvement, moderate improvement, mild improvement, unchanged. Such arbitrary classification should always be avoided. Redness, absent, mild, moderate, severe, swelling, no swelling, feeling of swelling, heaviness, apparent swelling, huge swelling. See, these are all very arbitrary terms. Better always be avoided. Always go for some standard, internationally accepted criteria for evaluating any clinical condition. Also, don't use names or abbreviations of the names of the patients. Don't use their pictures that may disclose their identity. Don't use pictures or artwork without prior permission. So these are certain important points to be taken care of while writing the results. So last part of the paper is discussion, right? Under IMRAD format, the D stands for discussion. In discussion part, you should write what do your results mean in the context of the existing knowledge. How does this fit in with what other researchers have found? What are the perspectives for future research? So these are the three questions to which you are supposed to give answers in the discussion section. State the main findings of the study. This is how you write the discussion, right? Start with the statement of main findings of the study. Then discuss these findings with reference to previous research. So, are the results similar or are they different? If different, why are they different? How are they different? Ensure that the interpretations match with the data. Discuss the implications of new findings. If they are different, what are the implications of your findings? Analyze the strengths and accept the limitations of the study. Then offer the perspectives for future studies. Ensure that the data is adequate to support these conclusions, and also accept the limitations of your study. The references are out of date and obsolete. This is a very common problem that is encountered. The discussion does not provide an answer to the research question. Discussion overstates the implications of the results, and discussion often does not acknowledge the limitations of the study. So these are very commonly observed lacunae. The last section after results and discussion is the references. So when writing the references, you should keep in mind certain facts like the functions of references, right? References serve three functions: to give credit to the previous workers, to add credibility and authenticity to your statements, and to help readers find further information. These are the three functions that all references serve, right? One of these functions are always served by a good reference. Contribution of a paper to the science is often assessed on the basis of citations the paper receives or the author receives. Citation management like EndNote, Reference Manager, Citation Builder or Mendeley are available and can be used to ensure the accuracy. So while writing the acknowledgement section, thank all those who helped in your study, maybe the volunteers or funding agencies, and those who provided you laboratory facilities, or patients who agreed to receive your treatment knowing that it might also simply be a placebo, and anonymous reviewers or reviewers of other journals where your article might have been rejected from. All these people may be acknowledged in your acknowledgement section. And with this, we conclude this lecture on the parts of a research paper. Thank you for patient listening.